presentation on the challenges in drug discovery and development at the preclinical stage. The first concern that I will discuss about it is how to isolate a drug and how it used to be traditionally, how it used to be handled. And traditionally, what we used to do in order to discover a new drug, our source was nature. In other words, we used to be able to isolate active principle from plants. And of course, when we identify compounds that are active, we could, using chemical synthesis, to be able to synthesize these particular drugs. So, field certainly called natural products. Natural products. So, synthesis of natural products used to be a strategy to achieve the synthesis or to achieve to develop or discover drugs from originally a lead compound identified from a plant extraction. So structural modification of these particular natural products could lead to a wide variety of structures which could be screened in order to identify potent drugs. However, the development of these strategies limited by the choice of targets that was a little bit scant at the time, traditionally, and importantly, now in our current era, producing compounds are extremely, I wouldn't say extremely easy, but we are tremendously aided by our ability to generate small molecule libraries, and also, more importantly, in terms of target, we are no longer as limited as we used to be for the past few decades, as now we have identified a number of genes that are directly associated to diseases, most particularly in cancer. And the use of structural methods for detailed characterization of ligand-protein interaction sites have significantly aided the development and identification of new drugs. Now, of all the parameters, the most important advantage that we gained over the past decade is certainly the increased knowledge of cancer biology. So we now understand the importance of the tumor microenvironment for tumor development, and we have identified the targets that can be modulated by both factors released by the tumor microenvironment, and we also understand now much better the molecular basis of metastasis and invasion. So now, in this modern era of cancer drug discovery and development, we are significantly advantaged over when compared with the past 3 to 4 decades. We have achieved the synthesis of new drugs based upon 2 major strategies now. We have a number of strategies now that allow us to design, to be more creative in terms of drug discovery. The first method is what we call the ligand-based design. The ligand-based design is defined as a strategy based on the knowledge of the ligand. In other words, the knowledge of the bioactive agent. And we also have the structure-based design as well, which is a drug design strategy based on the 3D structure of the target obtained by X-ray or NMR. Now we can even co-crystallize an inhibitor or a ligand with its cognate receptor or its cognate target. So in the next slide, I will show you an example of the co-crystallization of a drug called Tarceva, which was designed to target EGFR, epidermal growth factor receptor, slide number 5. So you can see in slide number 5, this is the X-ray structure of co-crystallization of Tarceva with EGFR. As you can see, the molecule binds to the ATP pocket in a way that the phenyl ring is anchored into a hydrophobic pocket, and the pyrimidine type of ring in the quinazoline system is forming a hydrogen bond with the receptor. So we know well by using co-crystallization, we can easily define the critical parameters for a particular molecule to bind to its cognate receptor. So this is an enormous, a clear example of the technological advantage that we have in terms of creation of new molecular entities. Now, it is technologically feasible for many settings to be able to design a drug, either model it using molecular modeling, and also ultimately co-crystallize the drug in order to be able to define critical parameters and the critical function of the molecule that can underline the binding of the molecule to the cognate site. So we have achieved a lot in terms of methodologies, in terms of strategies to successfully create or discover a new drug. However, as we will see in the next slide, the rate of failure is still extremely high. The percentage of new cancer drugs that are approved for clinical use or that are clinically approved by the FDA is only 5%, which means that most of the drugs of the drug candidates fail in the clinic, and mostly due to the lack of efficacy. So I should say here that despite the fact that we are very advanced in terms of our understanding of cancer biology, despite the fact that we are very advanced in terms of tools for drug design, in terms of assays for evaluating drugs, the rate of failure is still very high. As I repeated again, only 5% of new cancer drugs are approved by the FDA. So questions arise as to why, despite the modernity of our technologies, we are still failing in the clinic. So there are, next slide please, slide number 7. There are some arguments to explain why the rate of failure, what we call the attrition rate, is so high. Well, it is believed that cancer is a very complex disease, which is characterized, as I mentioned before, by the tumor microenvironment. So the tumor microenvironment is extremely complex. As I mentioned before, the tumor microenvironment can be forming growth factors that the tumor can use to proliferate, or can also use to activate expression of genes associated with invasion, thereby promoting tumor progression. Now, another parameter that is extremely important in terms of complexity of tumors is the intra and intertumor biological heterogeneity. Heterogeneity is one of the major factors that complicate our strategies in terms of designing molecules that can block tumor proliferation and progression. Now, there is also another barrier to drug discovery, to effective drug discovery against cancer, is of course the systemic and tumoral immune and metabolic response heterogeneity. So drugs which are designed most of the time to block one simple target are not able to modulate the immune system when they are, for example, designed and synthesized to target DNA or to target any other metabolic proteins that are involved in DNA synthesis. So the aspect that we always miss when we design a drug for a specific target is always the immune system. Now we are seeing now drug development and drug discovery directly directed at the immune system, which are the class of drugs that we now call the immunomodulators. So number 4, what we can say also is the ability of the drug-resistant stem line following therapy to be able to repopulate the cancers. And these particular stem-like cancer cells are capable of causing tumor regeneration. So if our drugs, they fail to kill this particular stem-like cancer, we have, as a result, the tumor research with the tumor, we can see that the tumor can be repopulated and again started to progress again. So with a population of tumor cells that are usually much more resistant to the primary therapy used to control this tumor progression. So cancer is an extremely complex disease, and as you can see, I only described here for the sake of time only 4 parameters that outline the complexity of tumors. Therefore, these parameters are very rigorous and very solid barrier to anti-tumor therapy when we are trying, most particularly, to target new oncogenes that have been identified. So, despite the fact that we are able to design new drugs, we are capable of inhibiting at low nanomolar level some particular targets, such as, for example, the EGFR, the epidermal factor receptor. Yet, when the tumors are treated with a single drug, they are bound to fail and they are bound to generate resistant population and thereby progressing further. So, why do we fail to address these particular disease complexity? Most of the problems are really in our preclinical model. So our preclinical models are usually designed in mouse, in mouse system, and in models that are developed in the mouse, and some in the rats, but generally the models are in the mouse. We do not have other alternatives to the mouse, and one of the reasons this is as a result we have been able to improve the models, to work on improving the current models that are available. So I will give you a very short review of a very short overview of the currently available model, and then as you will see, as I will talk about these models, you will see that we have tremendously progressed towards trying to develop models that recapitulate well the problems that we just discussed before. For example, the tumor microenvironment, the heterogeneity, and also a proximity or a greater similarity, a greater simulation of how the drug can be in human and how human tumors can respond clinically. So there is a barrier between the mouse and human, as you will see, and we are bound to develop our models in the mouse. So before making comments about the challenges that we have in this preclinical phase of drug development, I will describe these particular models, and I've chosen to talk about 4 of them. And the first one, next slide please, is what we call the syngeneic model. The syngeneic model is such that, okay, here is the list of models. We have the syngeneic model, mouse model. We have the cell-derived xenograft that is called CDX, and we also have the new patient-derived xenograft, which is commonly called PDX, and we have also the hollow fiber assay. This particular assay was developed at the National Cancer Institute of the United States, and it allows to rapidly evaluate the potency of compound in vivo in human tumor cells implanted in the animal. So I will describe this particular novel and advancement in terms of in vivo model for drug screening. Finally, we have the genetically engineered mouse, and this is called the GEM model. And in these models, as I will show you, these models are designed for the benefit of this model is such that the tumor can be at its initiation stage and also at its progression stage, which is a better mimic to tumor formation in human. Next slide, please. So let's talk about the syngeneic mouse model. What is a syngeneic mouse model? Is that the cells are the tumor cells that will be used to test. Voila, nice. Thank you. That will be used to test the drug. They are mouse cells that are implanted in the mouse. So that's why we call it a syngeneic model. So traditionally, over the past, I would say 40 years, the syngeneic models were very often used. The classical leukemia P388 model, and some of you may have heard about this particular model. There are mouse cells that are implanted in the mouse, and then the drugs were given IP. Now, the next most advanced, more advanced model is the xenograft model, the cell-based xenograft model. And in that particular model, as you can see, the mouse that is represented here bears a tumor on the dorsal, part of its body. And the tumor mass that you see here is made of human tumors, of human cells. In order for the mouse to be able to tolerate the human cells, you can guess the mouse must be immunodeficient, which is a disadvantage in terms of mimicking or recapitulate human tumors. The human has a robust immune system, but in our model, the mouse is immunocompromised. The next slide, please. The next slide will depict the type of results that my laboratory has obtained using the xenograft model, which consists of grafting human cells onto mouse that are immunocompromised. Just to give you an example, we were developing, we're still currently developing in our group, a concept called combi targeting. This particular concept is taking into account the complexity of cell signaling that control tumor progression, and in the sense that we wish to design molecules that are not single targeted, but rather multi-targeted. So in this model that I'm showing you here, we designed a molecule to be able to generate another molecule capable of inhibiting a target called EGFR, the epidermal growth factor receptor. But the molecule is also programmed to generate what we call a methyl diazonium. And this particular methyl diazonium is an alkylating agent that is designed to damage DNA. So therefore, this particular molecule is designed to be an inhibitor of the epidermal growth factor receptor, and also to behave as a cytotoxic, anti-cancer drug, a DNA reactive drug at the same time. So in other words, the molecule, as small as it is some kind of a hidden combination, or a masked type of combination that kind of establishes a bridge between traditional chemotherapy by targeting DNA and the modern targeted therapy by targeting this kinase ATP domain of the EGFR. So when we wanted to make the proof of concept of this approach, after elucidating the in vitro mechanism of action of the class of drugs, we have developed some optimized structures, and we decided to go into the preclinical phase, and we decided to evaluate the potency of this compound in a model that recapitulates not totally, but at least approximate human tumor growth. So we used the xenograft model where we implanted the human tumor cells. In this case, it was we implanted two human cell type. One cell type, which was next slide, please. One cell type, which was prostate cancer cell type, and the other one was a breast cancer cell type. So in this model, what we can see is that on the y-axis we have the mean tumor volume, and on the x-axis we have the time. And as you can see, the time in days. So the study lasted 25 days, and you can see that we have a control. The control is the graph on top. We can see that this particular drug, we call this new drug combi molecule, at a 100 milligram per kilogram dose. We could see that this particular drug blocked the progression or the proliferation of these particular cells within the tumor. So by reducing the tumor volume as a function of time, we could predict that perhaps this particular molecule could behave in human as a potent anti-tumor agent. So the next slide I will show you, these xenograft models are expensive models, and when their cost can be up to, for some studies, can be up to $40,000 when it is done by CROs in the industry. And in academia, the cost is, of course, less. But however, they are extremely important model that is used in the pharmaceutical industry and also used by what we call the CROs the contract research organizations. So I will talk to you now about I would say that an example of an asset that was developed in a government institution, the National Cancer Institute of the United States. And this particular assay is a very versatile one. It's an in vivo assay called the hollow fiber assay. It consists of taking fibers and fill these particular fibers with human cancer cells and implant them SC subcutaneously in the animal. So on the left side of the slide, you can see an example of an animal to which one of these fibers was implanted. And in this particular fiber that you can see here, the cells, in order to verify whether the cells remain alive or remain viable, the cells were transfected with luciferase. And luciferin was given to the animal. And we can see that, unfortunately, it's not a color picture. We can see that, the fiber was shining on the imaging, indicating that despite being inside a fiber, the cells remain alive, and also the cell being alive in vivo when they are implanted to the animals. So the advantage of this assay is that it allows in vivo to be able to utilize, for example, three types of tumor cells. One fiber can contain lung cancer cells, and one fiber can contain, for example, brain tumor cells, and the other one could contain brain. So therefore, we could test one particular drug on three different types of tumors in this very versatile model, which was invented at the National Cancer Institute. It is called the hollow fiber assay. So as we'll see, with the next slide, please. At the end of the assay is a very short one, and after 7 days, we can sacrifice the animal, and we take the fibers, open the fibers, and we pour the fibers into 12 well plates, and ultimately study using dyes like MTT, ultimately study the growth of this particular drug. So if indeed the drug was capable of blocking the growth of the tumor cells, when they are transferred to the plate, their growth will be delayed when compared with the control. So therefore, it's an extremely important preliminary assay that has allowed the National Cancer Institute to screen drugs that can go into the xenograft model that is a much, much more expensive model. So since the mandate of the National Cancer Institute is to screen and evaluate a large number of drugs, this particular hollow fiber assay is still and was extremely useful in terms of pre-screen. Important parameter for this particular assay is the fact that when the fiber is implanted SC, the drug has to have appropriate pharmacokinetics in order to be able to deliver to this particular fiber. So it is also a test of the drugability of this particular structure, whether the structure can be really absorbed and be available systemically. So the hollow fiber assay is really a very versatile one, which allows to rapidly discriminate, rapidly rule out, rapidly remove from the progression path any drug that shows inactivity for 1 or 3 or that show that there is an equation, a threshold of activity, which I do not have time to go through, that is used to gauge the potency or the acceptability of the activity of the drug. So the score is a score of 20, and if the drug can score 20, it will be allowed to be used in the more complex xenograft at the National Cancer Institute. Now, the next form of preclinical model that is used very often, and that is now a revolution, that is the closest, we believe actually, to be the closest to a capitulated human tumors, is one in which we're still using the xenograft, where we take the tumors directly from the patient and then graft the tumor into the mouse that is, of course, immunocompromised, and then allow the tumor to grow. And if the tumor is aggressive enough, or if the tumor is capable of growing in the mouse, it will be possible to collect this tumor and then implant it to 3 other mice, thereby being able to expand the number of mouse with mice with xenografts for further studies, particularly studies with drug testing. So here is an example I cannot point. where we can grow the tumor in an F, we say F0, F1. If it's F1, the tumor grows into the animal, and then the tumor is collected. It's a fresh tumor from patient, and then the tumor can be implanted to treat other mice. And the mice can be used to test the drug. As you can see from the graph on the right, you can see the green, the blue graph that represents a drug X, an unknown drug, and you can see the vehicle. The vehicle is the solution that does not contain the drug, the solution that is used to dissolve the drug. So as you can see, if X is tumor volume, and then is Y is tumor volume, and X, as in the xenograph, is time in days, you can see that if the drug is effective, it will prevent the growth of the tumor, that is of the mouse with the tumor that is given the vehicle. So therefore, as you can see from this cartoon, you can see that the drug indeed is being tested on practically fresh tumors from patients, and then the tumors in the first passage can be benefiting from the stroma, which is the human stroma instead of the mouse stroma, which again is getting us closer from to the situation of the tumor in human being, in human. So the PDX xenograph is the PDX patient-derived xenograph is an extremely versatile model, extremely clinically relevant, and the industry of xenograph now is twofold. One is to be able to follow patients with their own xenograph their own tumor being implanted in mice, and the other one is to use these particular mice to be able to test the drug. It is expected that by 2022, from a source from the internet, that the market for PDX in the industry of CROs and others in the pharmaceutical industry, it is expected that this industry will reach $167 million by 2022. So it's an extremely promising and extremely versatile model to recapitulate human tumors, and of course to give us really one of the closest proximity, the closest reality of human tumors with the tumor microenvironment and so forth, in order for us to define whether a particular drug deserves to be moved at the subsequent phase of drug development. Now, next slide, please. In the next slide here, I will talk about one of the few challenges of the PDX. Although the PDX is a very versatile model, there are some limitations. For example, please go back. There are some limitations. Some limitations of the PDX is the time it takes for the tumor to take. Sometimes it can fail. If the tumor is not aggressive, it may fail. However, even if the tumor is, there is a long time for the tumors to take. Sometimes average 2 months, and sometimes it can be even longer. Okay. However, the PDX has the advantage to individualize precision cancer care, and it allows to, in some cases, there have been examples where one could find a particular drug that could be potent against the tumor using the model. The drug that is, of course, an already approved drug can be used in human being, and it has already been shown that drugs identified in the PDX model can be effective in human tumors. Now, there are other limitations that I should mention, is that the cost of the PDX is enormous, and certainly there is an unpredictability of the time that it will take for these tumors to take in the animals. So advantages and challenges, of course. The PDX model is a challenge. The challenges are being resolved progressively, and one conclusion that we can make very rapidly about the PDX is that it recapitulates the reality of the tumor milieu in human, and also allow to test the drug in a system that directly come from the patient, fresh tumors directly from the patient. Then genomic profiling of these particular tumors are recapitulating as well the profile of the tumors from human. Now, next slide, please. One of the models that I must mention to you here is the intracranial model in glioblastoma and brain tumors. So, as you can see from this slide, brain tumors are extremely difficult to model, and for the simple reason that the blood-brain barrier is a significant obstacle in terms of tumor distribution, in terms of course, you can say in terms of the therapy of these cancers. So it is critical to find a model that is not necessarily implantation of xenograft SC subcutaneously in animal, because if we do xenograft, if we perform xenograft models SC in the animal, we will not have the critical human barrier, which is the blood-brain barrier. So we have preclinically to develop models to recapitulate the challenges that are presented by the blood-brain barrier in brain tumors, glioblastoma, for example. So how do we do that? We can, in an IC intracranial model, implant the tumors into the brain of the animal with an extreme caution. We're using special equipment for tumor implantation to implant this particular tumor without killing the animal. So here an example that we have recently successfully performed here at our institute is an example of a tumor that is resistant to a drug called Timodel. And this particular example shows that the tumor, as you can see, and later on, since these particular tumors, the tumor cells, they are transfected with luciferase, and they are given luciferase, and from the process of luciferase, they become luminescent, and we can follow thereby the tumor in the brain of the animal. And you can see I'm giving you here a very typical example where you can see that the tumor can progress in the next in the first row is six. The first row represents 6 mice implanted with the tumors, and then 6 days later, we can see that the tumor significantly progressed, as the image is showing, in the brain of the animal. So this particular model allows to test drugs with the complicated component that the blood-brain barrier represents. So these models are extremely expensive in terms of the industry of CRO, and they are not models that are easy to perform. The implantations are extremely delicate. Otherwise, the animals are going to be killed. Given the cost of the animals, it's extremely important that the implantation be successful. So this barrier, as you can see, of modeling brain tumors in the mice is at least partially solved by using the IC, the intracranial implantation model. However, there are limitations to this. Is that it doesn't really follow the scheme of tumor progression that make it a different and more faithful mimic of tumors in human, or brain tumors in human. Now, an important challenge in preclinical is, although we have now models, which we call models to represent the pharmacodynamic of drugs, it is also important to consider the pharmacokinetics of the drug, and as we will be shown by the next slide. Can you show the next slide, please? Next slide. And again, before we go on to talk about the pharmacokinetics, we can summarize about the challenge of the pharmacodynamic models that we have. Number one, there is a lack of reliable preclinical technologies that can recapitulate faithfully the complex variables in therapeutic responses and diagnostic accuracy. These can also be one of the main reasons for the high attrition rate that we see in drug discovery and development as well. Now, the other parameters, a parameter which is also not to be, is the difference between human and mouse. although there are a lot of similarity between human and mouse, for example, in the immune system and so on, it is important also to consider the fact that some differences can, for example, can be elusive, can be confounding. For example, the mice, as I will say, the mice can tolerate higher doses of drug than human. And if we want to have an extremely faithful representation of drug activity in human, it is very important to consider the toxicity level, which is extremely favorable in mice, but not necessarily in human at a certain dose. Now, speaking about differences in mouse and human, let's look at the pharmacokinetics. Just before we talk about the pharmacokinetics and the metabolism of this drug, let's just have a quick look, please, at the next slide, please. A quick look before we move on at the genetically engineered model. The genetically engineered models are from the last slide I was referring to the immune system of the mouse, and they are the most compatible model for our current rapid development of immunotherapy and also development of immune checkpoint inhibitors. So that they are the best, the genetically engineered model, the best preclinical platform, because in this particular model, the mice are not immunocompromised. These particular models recapitulate initiation and progression of tumors. Now, although some immune properties differ from mouse and human, there is significant conservation between these two. And that is one of the main reasons why the great advances now of immunotherapy or immunomodulation therapy that is one of the reasons why the progress can be significant. And I mean by that the main reason is the availability of this particular model with, of course, the similarity, the conservation of the immune system when compared with the mouse and the human. Now, as I said before, the number 3 of this particular slide, I mentioned before that the major advantage that the genetically engineered, the GEM model is offering is that it recapitulates tumor initiation to tumor progression. That is a very, very important important mimic or simulation or emulation of how tumors are initiated in human and how they progress. So parameters associated with initiation and progression in the tumors are maintained in this particular model, in contrast to the xenograft model, whereby we just take the tumor cells from human and then graft it onto the animal. Now, next slide, please. We will now look at another parameter where really the differences in mice can affect predictability of clinical response. Now, just to kind of review a little bit the duality between pharmacodynamics and pharmacokinetics. If you look at this cartoon here, when the drug is administered, and when you can read the word absorption from the top of the slide, the drug is delivered systemically. It is in circulation. It's in systemic circulation, and from systemic circulation, it is delivered to the tissues. And within these tissues, in terms of in the case of solid tumors, the solid tissues are the loci where the tumors can be localized. In the case of metastasis, the tumors can be in many different part of the system, and of course, the drug need to be delivered to reach these particular tumors that have been in remote tissues, sometimes tissues that were remote from the original cancer or the primary cancer. And then on the right, you can see the drug metabolization. Drug metabolization is a critical parameter for the fate of a drug when it is administered to the mice. And in many cases, the enzymes that are responsible for the metabolization of the drug in the mice, they are different from the ones that are in the human. So the fate of we do not have any choice of any other models, but we have to be very cautious on making prediction when we use the mice as our model, because the metabolization can be different from mouse to human. Now, if you look into this graph, we have the clinical response, and the clinical response will be composed of two parameters, two very simple parameters. One will be the toxicity, and the other one will be the efficacy. So the duality of pharmacokinetics and pharmacodynamics will be about the clinical response, or in the mice, the tumor volume, whether the tumor volume, we have reduction of the tumor volume or not, which is what we have talked about initially in all this model. And then the pharmacokinetics, where we study the kinetics of distribution of this particular drug in circulation, and also the duality between circulation and the systemic distribution and tissue distribution as well. So the systemic circulation, and the tissue distribution as well. So drug metabolism is important. Differences in the mouse can affect the predictability in vivo. Now, please, next slide. Now, I have presented to you now challenges in terms of pharmacodynamics, challenges in terms of pharmacokinetics. All these challenges are bound to the fact that mouse are different model than human, but we do not have a lot of choices. Most of our preclinical models are carried out in the mouse, whether it is in industry, whether it's in academia, whether it's in discovery in academia. So important parameters to be considered, which are also challenges. Number one is the pharmacokinetics, the ability to modify the targets, which is PD, which is the pharmacodynamic. Pharmacokinetics is the ability to study the kinetics of distribution of the drug systemically, and pharmacodynamics is the ability to modulate the target. And this, of course, we advise for this challenge to perform them in the tumor-bearing mice. And usually, classically, what we do is that we study our pharmacokinetics in non-tumor-bearing mice, and then we study our pharmacodynamics in tumor-bearing mice. So it is now recommended to enhance predictability that we do the pharmacokinetics in the mice that bear the tumors. Now, other challenges, the fate of the administered drug, it is largely dominated by absorption and metabolization, and distribution, and excretion. So what's the so-called ADME? So thus, the phenotypic differences between the central metabolizing enzyme in mice and humans that are different, they represent a confounding factor in extrapolating drug PKs from mouse to human. So I think I have repeated that enough as a caution to be as a parameter that can be significantly confounding in terms of predictability of drug effects in human. Now, finally, importantly, as I mentioned before, the MTD, the maximum tolerated dose of many drugs in mice, is significantly higher than in humans. So it is essential to evaluate efficacy by using doses achievable in patients. And it is some kind of a complicated challenge because when we are developing a new drug, we do not have the concentration, the achievable concentration in patient available. So we have to rely on the mouse and make significant calculations to be able to arrive at the first dose that we can bring to human. So this is a major challenge. In the case where the studies in vivo are being done with a drug that are being performed with a drug that is already in the clinic, we are guided by the clinically achievable concentration. But in the case of a completely new drug that is being discovered, we do not have this guidance from the plasma concentration available known from the literature from previous work in human. So next slide, please. I will in this very next slide talk a little bit about the path toward the bench to the clinic and where the preclinical is situated. Now, in drug discovery, we start by, of course, having a target, by selecting the agents, by doing in vitro work, by doing several in vitro work to be able to demonstrate that the drug has the mechanism of action that it was designed for. And then we determine in vitro assay. Later on, if the drug shows vitro in human cells drug, if you consider that the drug is more potent vitro than the standard of care drug, then we advance the drug to the models that I just discussed. And remember, these particular models are very, very expensive. So it is extremely vitro. The drug shows that the drug that is selected from the stage that I labeled here, stage 2A, it is extremely important that this drug is optimized. It is the conditions have to be set so that the identification of this drug is completely unequivocal before embarking into extremely expensive vivo studies. As you could see, the extremely expensive, vivo studies. So as you could see, the syngeneic models are the least expensive because they involve putting mouse cells into mouse that are not necessarily expensive. But once we move towards the models of the xenograft model, the mouse costs much more. They are in the range of $100. And if we're using 60 mice as a rule, you can already make the calculation and then guess how expensive it will be. So therefore, it is extremely important to select an optimized drug, and then assay for this drug or detect or study this drug in vivo. As you can see, the identification or the demonstration of the effect of this drug in models that are clinically significant certainly can give us a more confidence, a greater confidence of the potential activity of this drug in human at a later phase. So therefore, as you will see here in this case, the preclinical phase, the preclinical pharmacodynamic evaluation, is an extremely important one. Now, not to neglect, in stage 2B, as I labeled it here, the pharmacokinetics. So the pharmacokinetics of the drug is important. We have to be able to demonstrate that the drug has reasonable bioavailability. By this, we mean that the half-life of the drug is reasonable when compared to drugs of the same class. We have to be able to demonstrate that the drug is stable. We have to be able to demonstrate that the metabolization are not deactivating the drug, and that indeed the level of metabolization keep the bioavailability of the drug with an adequate level to be able to be delivered to the tumor. Now, the next stages are the stage number 3, where the drug has to be evaluated in preclinical toxicity with the dog and the rat. The dog and the rat, these level of toxicity is called GLP toxicology, and the GLP toxicology is extremely expensive. It can go all the way up to $500,000, depending on what is required in order to advance the drug for an IND and later on for phase 1. So I'm putting these steps here so that I can highlight the importance of the challenges that we have in the preclinical stage. First of all, we have a predictability challenge. We are using mouse model, and we have no choice but to use a mouse model. And then second of all, we have to move the drug further if the drug is shown to be potent or satisfactorily potent in this particular assay. So we have to show that the drug are tolerable in GLP, good laboratory practice toxicity, which is an extremely costly phase of the drug development. And the cost and the challenge or predictability are in fact major obstacles at the preclinical stage of drug development. And later on, in the phase 1, the phase 1 is a little bit less risky, but however, finding funding to do phase 1 is another challenge in the industry. And the phase 2 is enjoyed for a drug to be moved to phase 2, but however, the activity of the drug can be proven to be not to the level of standard of care drug, and unfortunately, these might lead to decision to stop the progress of the drug. As I mentioned before, only 5% of the drugs that are submitted to that level are finally, ultimately approved by the FDA. So finally, conclusion to all, and as I mentioned that it was going to be a very short presentation. So next slide, please, conclusion. So it takes an average of 12 to 15 years to develop a drug, and an average of 1 billion for the drugs to be moved from the bench to the clinic. And within this path, we are facing major challenges. And preclinical years today was the topic which was the topic of today, is an extremely important phase. It's a critical phase to be able to decide whether the drug should be selected for moving to the next phase. And in order to improve our predictability, we need models that better recapitulate human cancers. And we are working at it. I consider the PDX, the patient-derived xenograft, to be a significant advancement in terms of recapitulation of human cancers, human cancers. And needless to emphasize, the effort towards reducing the cost of the most advanced model. A syngeneric model is not very expensive. However, from the xenograft to the PDX to the genetically engineering models, we're talking about extremely expensive cost, extremely high cost in order to be able to test drugs and define finally whether there could be potential effective new drugs in humans. Now, the good news here is finally is that we have hope. The hope is based on the fact that the tools for drug discovery and development are being updated regularly, and as we can see, we are progressing significantly on number one on the technologies to be able to implant human tumors in the mouse. We are progressing significantly in the genetically engineering models in such a way that now we can test the immunomodulators. We can test PDL1 inhibitors. We are also progressing significantly in the screening, as I've given you an example of the hollow fiber assay, where we can test one particular drugs against many tumor cell types, and finally have a threshold criterion in order to be able to select the drug to advance into the most expensive phases. So we have strategy to reduce the cost as well. So thank you for your attention. And again, I apologize. I think there was a cut in the communication, and I lost you for a minute. So thank you very much for giving me that opportunity to be talking about the preclinical models and their challenges as well.